Hi, my name is Warlaumont, and I am presenting this journal track presentation on behalf of myself and my co-authors. The first author on this work is VPS Ritwicka. She is the person who did the majority of the work for the paper, of course, and she's the other corresponding author on it. And then our collaborators were Gina Pretzer, Sarah Mendoza, Chris Shedd, Chris Kello, and Ajay Gopinathan. And this is a paper that was published this year in Scientific Reports, and it's called Exploratory Dynamics of Vocal Foraging During Infant-Caregiver Communication. So the motivation for this work is work on foraging as you probably normally think of and use the word. So there's been a bunch of work, a body of work, that has studied animals' movements basically on map in a 2D space, characterizing their movements in terms of these discrete periods where they're collecting something versus wandering in search of something else. And you can take those periods of wandering and look at how long they took in time or how far the animal traveled in space. And if you do that and you collect a number of these measurements of durations or distances traveled, you can create what are called step size distributions and characterize those step size distributions, for example, by fitting different types of curves to them. So some research has found that by seeing whether power law or log normal or exponential fits are better fits to the data, it can be very interesting because in some cases the type of fit that you get as best fitting the data depends, or it seems to have a relationship with the scarcity of resources. And others have done some work modeling how different types of search strategies would associate with an animal's success in obtaining the resources that they need to survive and thrive, and in different hypothesized distributions of resources. This is an image from one of those many papers on this topic by Humphreys et al. In 2012, studying albatross behavior. Albatrosses have been a very important animal in this field, and this is a book called The Physics of Foraging by Viswanathan and colleagues that provides a nice overview of this kind of approach from the perspective of a physicist. This type of looking at behavioral data has also been used to study foraging by humans in non-spatial domains. And so our collaborator on this, one of our collaborators on this project, Chris Kello, has done work studying foraging in memory, where the space is not a physical space, but is instead a semantic space. So when he and his collaborators have participants come into the lab and try to recall as many different animal types as possible, for example, they find that the inter-response intervals that human participants produce seem to have potentially power law dynamics to them, the way that we see sometimes in foraging in space. And they also find that the path length, that those inter-response intervals, that's shown here on the x-axis, are correlated with the distance between two consecutively retrieved items in a semantic space. So what happens is basically participants will they'll retrieve a cluster of items that are similar to each other, like maybe a group of farm animals, and then they'll pause for a while, and then they will discover another cluster, maybe like aquatic zoo animals or something like that. And that they do this in a way that's that that where the clustering both in time and space seems to be, wow, so I'm already taking a really long time here, so I'll really have to condense this. Okay, so we have some evidence already from the same kind of data that I'm going to talk to you about today, because they're the focus of this recent paper. So these are data collected using the Lena system for creating long recordings. Usually they're in our studies at least 10 hours long, but they can be up to 16 hours long. These are recordings that are collected by parents on behalf of us as researchers, and they're done in the baby's home. And the baby wears this, usually a vest or another clothing item that contains the recorder for the whole day. And then the Lena system does also include some software that allows us to automatically identify when vocalizations of different types are occurring, in particular when the child is vocalizing, when adults are vocalizing, and whether when the child's vocalizing it's a cry or a speech-related vocalization like a coo or a babble or speech. So this is just an example of a figure from a paper by Drew Abney and some of us from 2016, where we were documenting the fact that vocalizations appear to produce both by adults and by infants, and infant vocalizations of both the speech-related type and the non-speech-related type, which are mostly cries. These are all clustered in these nested clusters over the course of the day. So that is what that paper documented. And what we're doing here, it could be in part thought of as an extension on that, beginning to look at infant vocalizations in this way. And we're asking, do infants and adults forage in an acoustic space? And if so, do social responses function as resources as they're doing this foraging? So for this study, we actually had two data sets that we included. And so I'm going to describe here the data collection and pre-processing steps. So we used the Lena system to collect day-long audio recordings, where we were asking parents to target at least 10 hours of audio recording in one of their recording days. We had two children who were recorded frequently, approximately 2 times a week over the first year of life, actually more like from 1 to 2 months to 13 to 14 months of age. And then we had 15 infants who were intended to be recorded longitudinally at 3, 6, 9, and 18 months of age. But at the time when we did the data analysis for this paper, we were in the midst of ongoing data collection. So we currently actually have more recordings from this data set, and we're planning to use those to do, hopefully, a replication study. We use Lena's software for automatically identifying when infant and adult vocalizations took place, and for separating pre-speech vocalizations from cries, laughs, and vegetative sounds. So we're very reliant on the Lena system for this processing to make it possible to look at a lot of vocalizations over a lot of hours, over a lot of participants, over a lot of days. And we used Pratt software to automatically estimate the pitch and the amplitude of each of these child and adult vocalizations. This then allowed us to use the onsets and the offsets of those child and adult vocalizations to identify when there were vocalization to vocalization steps. So we're considering something to be a step in this, abstract sense of what a step could be. If you have, or what we're considering as steps are consecutive infant or consecutive adult vocalizations. And then we're considering 2 different types of steps as well. So those that are occur with response and those that occur without response. A with response is an example. Here's an example here of an adult step that is with response because the adult vocalization, the first adult vocalization in the sequence is followed within a very short period of time by an infant vocalization. We set a 1-second cutoff for these analyses. So we're saying that the adults likely got a response from the infant when they vocalized, and then we're going to look at what's the amount of time that passes between this first vocalization and then the next adult vocalization that the recording picks up. And we're also going to look at the acoustic properties, specifically the pitch and the amplitude of the two vocalizations and how different they are from each other. So we can look at adult steps that had an infant response to the first of the two vocalizations. We can look at adult steps that did not have a response. We can just exclude from our analysis adult steps that are so close together that we cannot assess whether it was with response or without response, because not enough time has passed for us to really say yes or no that first vocalization by the adult in the sequence did or did not get a response. We're just going to exclude that step. And then we can also do the same for infants. And this is an example here of an infant vocalization step to vocalization step that had not received an adult response to the first infant vocalization. And we do have other examples where the infant, of course, did receive adult responses. Okay, so that's this slide. And then from all that, we can get intervocalization step sizes in time and in the two acoustic dimensions for both adults and for infants. This is showing you here a plot of what those steps would look like in acoustic space, where the two dimensions of the space are pitch and amplitude. We normalize across the entire data set of all infant vocalizations in order to create these pitch axes and amplitude axes. And we log transform the pitch as well, just to account for the nonlinearity of pitch perception. And then this plot is showing you the movement. It starts here actually. The movement from one vocalization to the next, how long it took, and then how far the two vocalizations are apart in acoustic space. So you can see that this was 51 seconds between these two vocalizations, but they're actually pretty close together in both pitch and amplitude. And you can see that the infant is traversing this acoustic space over this portion of a recording in this manner. So we do the same thing for adults. This plot is not showing you the with response steps separately from the without response steps. It's just to give you a sense of kind of a visualization of the data in this way. And here is that data again showing adults in blue and infants in yellow for a small number of steps, and then just for a larger number so you can get a sense of how they're distributed. We can also look individually at the different acoustic dimensions. We basically created distributions of these step sizes in time. So intervocalization interval then in space. And we could look at the median of those median step size. So a shorter step size a smaller median would correspond to a smaller typical step size, which would mean either that the vocalizations are tending to be closer together in time, or that they're tending to be similar to each other in acoustically. We also could look at 90th percentile, just look at a different aspect of the step size distribution. And we also fit either a power law, a log normal, or an exponential distribution to the step size distributions, selecting which of those types of distributions we would use using a Kaike information criterion. And then we used linear mixed effects regressions to look at how those features varied as a function of infant age and whether or not the particular speaker in question got a response. So whether the steps were with response steps or without response steps. So this is a lot of different information that we're getting about foraging patterns. And we end up with tables that look somewhat like this, which I'm not putting up because I'll give you the main takeaways in the next slide. But I want to make a few points. So one is that in general, we found that the majority of recordings for any given step type were assigned by with our AIC the same best fit distribution type, which was really helpful from a practical standpoint because it allowed us to compare the parameters of the different distributions of a given type. If there's more diversity in the types of distributions that turned out to fit a step type, that would have made this a little harder. We would have had to take a different approach to analyzing the data. So anyways, we ended up with these tables that were kind of this kind of type, where we would have for a particular step size type, step type, we would be able to look like here, infant acoustic step size or infant intervocalization interval, like the how long the step took in time. And we could look for each of those at median, 90th percentile, and then 2 parameters of the log normal fit to that step size distribution. And we could look at whether those were varying in relation to age or in relation to whether the infant got an adult response. And we found that in some cases there was like good converging evidence in favor of those steps getting smaller, meaning or larger, with respect to response or age. So some of the noteworthy initial findings that we got from reporting the paper using this analysis approach are, first, that acoustic step size and intervocalization interval for both infants and adults were weakly but significantly correlated. And so this is exciting because it parallels a feature of foraging phenomena in other domains, which is a tendency for distance traveled to be correlated with travel time. And for foraging in physical space, this makes intuitive sense because we expect it to take longer to travel longer distance, assuming constraints on an animal's speed. But like when studying adult human foraging and memory, the fact that there's a correlation between time and space in our vocalization data makes it kind of consistent with the idea of vocalization being a foraging process in an abstract sense, in the way that human memory can be thought of as a foraging process. The second thing we found was that for both infants and adults, time between vocalizations was significantly shorter for with response steps, which is kind of a new way of saying something that's been around in the literature for a while. So this finding aligns with prior literature indicating that adult responses serve to reward and reinforce infant vocalizations. It also suggests that infant responses to adult vocalizations may function as rewards for the adults. We also analyzed the data in terms of how acoustic measures were predicted. Like the acoustics of the first vocalization in a pair predicted whether or not that vocalization was going to be followed by a response. And we found some intriguing trends, but I'm not going to get into that here. And then there were, third, we found differences in acoustic step sizes as infants got older. So we found that infants showed larger step sizes, with, which is suggestive of broader exploration in pitch as they got older, but they showed smaller steps in amplitude. And then adult steps got larger in both acoustic dimensions. So that we thought was an intriguing finding that would be interesting to follow up on. We have some pretty significant limitations that we're going to be working on in the next phase of research. So the most major, in my opinion, is our reliance on automatic labeling, which was basically essential for being able to analyze step size distributions meaningfully to have enough steps represented. But as we tried to interpret our results and write them up, we realized that it was really good. It's going to be really important for us to do a proper, systematic, large-scale human listener validation of these findings, because if there's a tendency for software to systematically mislabel infant vocalizations as adult and vice versa, that could potentially be a confound for some of our results, and we really want to not have that be something that continues to trouble us. So that's something we're focusing on for the next phase of the project. And we'd also like to get more nuance in the acoustic characterizations. Looking at just pitch and amplitude is kind of limiting. And but nevertheless, we are excited to share this with you and with the world, because we think the focus on intervocalization steps is a very exciting new way to characterize infants' behavior. So I want to say a few things about how this is relevant to the ICDL-EpiRob community. So one thing I would like to say very briefly is that I do believe this is relevant, the type of approach is relevant for computational modeling and developmental robotics. For example, we could begin to evaluate models' fits to human data on the basis of how behaviors are distributed within a real or simulated day. So this provides another way in which we can assess and evaluate computational models. And then the other thing I would like to say more generally is that you can, we've put a lot of the data from this talk on HomeBank, where we had human participant permissions, and I wanted to mention HomeBank as a resource because it's another project of mine that I think will be of interest to this community. So I want to mention that you can now get free access to day-long child audio recording data on HomeBank, which is at homebank.talkbank.org. And if you go there, you'll find that actually you do have to go through a membership process and do various procedures to make sure that you're following the necessary precautions in dealing with the kind of data that are in there and taking, but once you go through that process, you could get access potentially to a lot of very interesting data from a lot of different interesting populations. And I'll also mention that materials for our paper are looking at these infant and adult vocalization step size data. The materials, the code, intermediate data files, and so on are on OSF, GitHub, and in supplementary material, and you can see the paper for details. So I'd like to thank the funding sources for this National Science Foundation, the James S. McDonald Foundation, and the various undergraduate research assistants who have helped us with this, as well as Kim Mahler, Tim Shea, Teo Rhodes, and Brian Kerser for assistance with various aspects of the project, and the infant participants and their families. And I'd like to thank you for your attention and for, for having this at the conference.